Welcome, online listeners and viewers, and thank you for joining this Science AAAS webinar, A Fresh Look at Fat Metabolism, studying lipoprotein lipase oligomerization dynamics with CryoEM and mass photometry. My name is Erica Berg, and I'm the Director and Senior Editor of Custom Publishing here at Science. I'll be moderating today's webinar. Joining us today as the guest speaker is Dr. Kathryn Gunn from the University of North Carolina at Chapel Hill, who we will learn about shortly. Finally, thank you to Refine for sponsoring today's webinar. And now let's begin. Blood fats are key biomarkers for heart, liver, and other diseases, highlighting the importance of lipid metabolism for health. Lipoprotein lipase is a key enzyme involved in hydrolyzing triglycerides from circulating lipoproteins in the blood, releasing free fatty acids. Understanding the structural and functional aspects of LPL is crucial, given its significant role in lipid metabolism and related diseases. In this webinar, the speaker will describe experiments that uncovered how LPL interacts with its triglyceride substrate and what governs its oligomeric states. Cryogenic electron microscopy was key to solving the structure of a dimeric LPL oligomer, while mass photometry facilitated a better understanding of LPL dimerization. Without further ado, I'm very pleased to introduce our speaker, Dr. Gunn.
Thank you, Erica, for that kind introduction. All right, so hopefully you guys can see my laser pointer that I've turned on. And yes, so I'm a postdoc right now at UNC Chapel Hill in Saskia Neer's lab. And I'm really excited to be telling you guys about the work I've done looking at the oligomerization dynamics of lipoprotein lipase. So before I get into it, I just wanted to start with a little refresher about why we care about lipases. So essentially, a large part of our diet includes the consumption of lipids. So these lipids need to then be transported from our intestine into our body so that we can use them as, functions. So basically, it's for different cell bodily functions, such as creating, such as energy or for storage for future use. And that's where lipases come in. You essentially can't bring a phospholipid or a triglyceride from your diet directly across a cell membrane. You need to break it down into its component parts. And so that's where lipases come in. They come in at every step of the process when moving these very hydrophobic substrates and break them down so they can go across membranes. And as you can imagine, lipase activity must be regulated. So the cell is actually made up of many different lipid depots, including a phospholipid membrane. And if you have lipase activity that's unregulated, that can be very damaging to the cell, since they can actually hydrolyze the component parts of the cell, such as the membrane. So understanding about lipase structure and dynamics and what governs these things is really important for better understanding how our lipid metabolism works. And so that brings me to lipoprotein lipase. So LPL is the lipase that exists in your capillaries, and it interacts with triglyceride-rich lipoproteins such as chylomicrons and VLDL. So these are packaged lipoproteins that have an inside triglyceride core. LPL can interact with these triglycerides to hydrolyze them and liberate free fatty acids. These free fatty acids, as I discussed before, can now be transported across membranes, and so they can head into the different tissues and be used as a source of energy in the oxidative tissues, such as like your heart or muscles, or they can be stored for future use in the adipose tissues. And we know that LPL is really important because without LPL, or if you have an LPL deficiency, triglycerides will build up in your blood. And you can actually physically see the result of that here in a patient blood sample from someone who has an LPL deficiency. You can see that the serum of their blood appears almost opaque, and that's from the buildup of lipids in the blood. Whereas if you have a patient with LPL that it's functioning properly, you're clearing out these triglycerides and the serum appears clear. So essentially, without LPL, you get this really big buildup of triglycerides. And so that means that LPL is essential for preventing hypertriglyceridemia, which is a risk factor for cardiovascular disease. And it's also important when it comes to pancreatitis, since hypertriglyceridemia can lead to pancreatitis. So knowing more about LPL structure is hopefully going to give us hints about how we could potentially treat hypertriglyceridemia and then also these resulting problems. So to start off with, I'm going to give you an introduction to LPL trafficking and structure. And that's because it turns out LPL trafficking and structure is quite complex in ways that we hadn't appreciated when LPL was first discovered. So LPL is synthesized in adipocyte and muscle cells. We know that once it's created, LPL is put into storage vesicles. These storage vesicles persist in the cell until a nutritional signal arrives for their secretion. And so this is one layer of regulation that LPL undergoes. After their secretion, the LPL enters the interstitial space. And we know that LPL has to then transit the interstitial space to arrive at the capillary endothelium. These endothelial cells are producing GPI-HBP1, which is a partner protein to LPL. LPL forms a complex with GPI-HBP1, and it actually facilitates LPL's transfer from the interstitial space into the capillary itself. Once it's in the capillary, LPL is then able to interact with chylomicrons and VLDL and work on its triglyceride substrates, liberating free fatty acids. We know that there's one further step in the process, which is that LPL can form a homodimer. This homodimer is known to facilitate uptake of remnant chylomicron and VLDL particles by the liver. And so if you look at these complex processes, when I started this project looking at the LPL homodimer, what we knew was a crystal structure of LPL with GPI-HBP1 and a CryoEM structure that I had solved of the LPL helical filament. And these two structures had certainly turned the LPL structure field on its head, because up until the point where these structures were solved, it had been thought that LPL was active as a dimer and inactive as a monomer. And these were sort of the two states that it had. And with these two structures, that idea was turned on its head. First, the structure of the LPL GPI-HBP1 heterodimer was solved. And as you can see here, you have a one-to-one complex where one LPL is interacting with one GPI-HBP1. And this was really surprising because we know that LPL can be active when it is in complex with GPI-HBP1. So suddenly the idea that LPL is only active when it is in a dimer doesn't make as much sense. Clearly it can be active when it's a single LPL with GPI-HBP1. And then on the other hand, the helical structure turned the idea that LPL is only inactive as a monomer on its head, because this helical structure is itself inactive. So LPL is interacting in the structure to form an inactive dimer that then that acts as the asymmetric unit of this helical filament. So from my point of view, the question that this really brought up was, what about these, active dimers? We had decades of biochemical evidence suggesting that they existed, but our structural evidence wasn't there. And so what I wanted to set out to do was to solve the structure of an active LPL dimer using CryoEM. And the reason CryoEM was really suited for this task is that one feature of these decades of biochemical data was that they had not been done in the presence of GPI-HBP1, which was only discovered in 2010. And so my theory was that the active LPL dimer existed when GPI-HBP1 was not there. And so in order to generate a sample of LPL that was homogeneous and did not have any helical filaments in it, I treated LPL with the bile salt deoxycholate. And this generated a homogeneous sample that we were able to freeze into cryogrids. And you can see an image of what that looks like here. My lab mates have often told me that this looks exactly like TV static, but I promise that if you take individual particles and average them together from within this static, you're able to see a signal. And that signal looks something like what you see here with a 2D class average. And this was really exciting because it seemed that my theory had been correct. If I treated this LPL with deoxycholate and froze it into cryo grids, we did seem to be seeing dimers. However, it also revealed there was maybe a small issue, which is that LPL was adopting a preferred orientation. So a preferred orientation occurs in CryoEM when protein groups at the air-water interface. So basically the idea is that after you apply your sample to the grid and plunge freeze it, the protein associates at the air-water interface. And the reason this is a problem is that if you want to do a 3D reconstruction of a particle, you need to have multiple different views of that protein. If you only have a top view, you're not going to be able to generate the entire structure. CryoEM relies on being able to see it from many different angles. And so I tried a lot of different strategies to beat this preferred orientation. And in the end, what we ended up doing is tilting the grid to find new views. So this essentially artificially introduces new views by tilting the preferred orientation away. And using this strategy, we were able to successfully solve the structure of our protein. And just to give you an example of what that looks like, you can see here our preferred orientation. And if we put in a 45-degree tilt, we now see a different angle of the protein. We also enriched for rare particle views using topaz neural network picking. And in combination, this allowed us to solve the structure of the LPL dimer at 3.9 angstrom resolution. And we were able to mostly overcome our preferred orientation. There is still some areas, you can see here with the sphericity rating of 0.846 out of 1. So 1 would be that you have an equal representation of all views from all sides of the protein. That we are, not at 100%, but we are doing really well. And so this then allows us to explore what we're seeing with this structure. And I had noticed in the 2D class that it did appear that LPL was forming what it looked like a tail-to-tail dimer. And this was really exciting to me as someone who studied LPL because it had been proposed that LPL would be forming a head-to-tail dimer in its active form. And this was the modeling that had been done previously, and no one had really suspected that a tail-to-tail dimer would be what would happen. But as you can see, the N-terminal LPL hydrolysis domain of one LPL subunit is actually facing the opposite direction of the other N-terminal domain, and the two C-terminal domains, which are involved in substrate recognition, are actually associating. And you can see I've colored the region of the C-terminal domain that's involved in substrate recognition in blue here. And hopefully you can see, as we look down the protein, that there is the active site located in the N-terminal domain colored in gray. So this arrangement, while very surprising, could actually make a lot of sense. But my first question was, why would this structure not exist with GPI-HBP1 present? And so if you just look at the two structures with LPL with GPI-HBP1 and then our LPL homodimer, and you map back the interacting residues for GPI-HBP1 and the other LPL subunit onto a single LPL, you start to see why that would be. And so it turns out they have overlapping interfaces. And so these two these two states of the protein are actually mutually exclusive. You could not have LPL GPI-HBP1 also forming the LPL homodimer tail-to-tail interface. So this is another look at the structure, but I've changed the thresholding so that we can look at the lower resolution data. So this is the data that we didn't resolve at high resolution because, generally speaking, it's mobile, or it's not staying in one place to allow us to actually resolve it. And the reason I wanted to show you this is that what you can see here is a density appearing, which I've colored in aqua, which is for the lid peptide. And for people who study LPL, this would be very exciting, but let me explain to you why. And so essentially, the lid peptide is the peptide that covers the active site. And again, the active site is in gray. And so in a closed version of the protein that is inactive, you would expect the lid peptide to be situated over the active site, since it's going to gate entrance into the active site. However, in this case, you can see that the lid peptide has slid to the side of the active site, revealing that the protein has undergone interfacial activation. And so this is what it's called when LPL interacts with a hydrophilic hydrophobic interface. And this is a common method of activation for lipases. So interfacial activation means that this peptide has moved to the side in response to the interface, allowing LPL's active site to be exposed. In addition, you can see the tryptophan loop, which is the portion of LPL C-terminal that is involved in substrate recognition, is also arrayed along the same surface. So you have the active site, the lid peptide, and the tryptophan loop of these two dimers arrayed along the same surface, and you can imagine how that would allow them to interact at the same time with a triglyceride-rich particle. The other feature of our structure that was very striking was that when you look down, you see that there's actually an empty spot in our structure, and it runs adjacent to the active site. And so this is back down to normal high resolution thresholding. The lid density, if you want to imagine it, would be located about right here. And so what I did was I characterized this empty pore, and I've represented that pore now as a green density. So this is the empty section running through the protein. And this did not really look, or this pore had not existed in the other two previous structures of LPL. So if you kind of look at the rearrangement of the residues in the center of the protein around the active site, you can see that there is a substantial difference between the crystal structure of LPL and then this structure of the LPL homodimer. So the main question this brought up for us is, are pores like this that run adjacent to the active site common in mammalian lipases? So to answer that question, we compared LPL to other mammalian lipases. And there's been quite a few structures of those solved using crystallography. And in this example here, what I'm showing you is two structures of pancreatic lipase that had both undergone interfacial activation, as we see that our protein has undergone. And you can see the lid domains of each of these proteins is in dark blue. So the lids of all of these proteins have then slid to the side, so they have undergone interfacial activation, exposing their hydrophobic binding pockets. So when we look at these two proteins and all the other structures of lipases that have been solved, we don't see that they have a similar hydrophobic pore. You can see that there is a pocket within which hydrophobic substrates can sit for these two structures of pancreatic lipase, and the active site is shown in red. But this does not look like what we're seeing in our structure of LPL, where the active site shown in red is actually adjacent to a hydrophobic pore that runs the entire length of the N-terminal of the protein. So we characterized the residues that are lining this pore, and that allows us to then look at the charge and the hydrophilicity of these residue of the pore at each section. So I've indicated the hydropathy here, which is a measure of hydrophilicity and hydrophobicity. So if you have a very positive number, that's very hydrophobic, so that's in yellow. And if you have a negative number, that's very hydrophilic, so that's in blue. And what I hope you can see is that when we start off here at the pore entrance, so this is the side that is next to the LPL lid. So this is where the triglyceride substrate would enter the protein. We have a slightly hydrophilic area. At the active site residues, the character of the pore inverts and becomes extremely hydrophobic. And we have a long stretch of hydrophobic section of the pore that stretches down to the end. And we can actually fit a monoacyl glycerol, so this would be a single monoglyceride, into the pore. And it fits very well. And so this made us wonder if potentially the pore was acting as not only an additional substrate discrimination site, so basically a way to help LPL specifically interact with triglycerides, but it could also be facilitating the movement of the acyl chain following hydrolysis out away from the chylomicron. So to illustrate this, I think we all have to think back to our days of studying an enzyme mechanism in college. And at least for me, when I think of how an enzyme works, I think of the canonical bidirectional mechanism. So we have an enzyme, it finds its substrate, it hydrolyzes its substrate into products, and then these two products enter and exit from the same direction. So it came into the pocket one way and it came out the same way. What we're thinking, though, is that it could be more efficient in the case of LPL to have a partially unidirectional mechanism. So following hydrolysis, instead of having the products exit from the same way they entered, you could have one product exiting from the other side of the protein. And this has the appeal of being very efficient because it would allow you to processively hydrolyze the free fatty acids without having to repeatedly release from the substrate. And, you know, of course, our first question was, well, is there anything that could be used to suggest that this might actually be a mechanism that LPL uses? And so we went into the literature, and there's a lot of LPL variants that are known to cause deficiencies. Interestingly, we found that 2 variants that are known to cause LPL deficiency localized to the bottom of this active site pore. So when these residues are altered in size or charge, LPL loses its ability to act on long chain substrates. However, in the case of the R102S mutant, it actually maintains the ability to work on small soluble substrates. So this does suggest that the LPL is folded and secreted, but that there is something inhibiting the long chain hydrolysis, or the long chain, the hydrolysis of long chain substrates. And perhaps it could be their release from this channel. So this is definitely an area that we're interested in studying more. To really, I guess, look at how this might work in a physiological sense, we decided to use Rosetta to model a triglyceride into this pore. And a triglyceride is composed of 3 chains. So usually they're called SN1, SN2, and SN3. And we know that LPL prefers the SN1 and SN3 chain of a triglyceride, so it preferentially will hydrolyze those two chains first. And so when we fit the SN1 chain into the pore that we see in our structure, the SN3 chain sits in this hydrophobic pocket. What we find very interesting, though, is that the SN2 chain is actually located in sort of a secondary pocket off to the side. And we do wonder if this could also be, if this basically substrate discrimination mechanism could be the reason that LPL prefers the SN1 and SN3 chains. Perhaps you need the SN2 chain to occupy this pocket in order to have the most efficient hydrolysis of the triglyceride. So, you know, we're very interested in our CryoEM structure, and we learned a lot from it. But one of the things that I couldn't shake was the question of why is LPL preferentially forming the dimer in our CryoEM structure? And the reason this, I guess the reason I was so interested in this is because of all that historical data that showed that LPL was active as a dimer. And so I wanted to rationalize that with our new knowledge of the LPL filament and then the LPL GPI-HBP1 heterodimer. So in order to look at the oligomerization dynamics of LPL, we decided to use mass photometry. So this is a really powerful technique that allows you to essentially weigh particles with light. So the idea is that you look at individual particle impacts as they hit a glass slide. You can then create a molecular weight calibration using known molecular weight standards to directly correlate the contrast you're seeing as these individual particle impacts hit the slide to normal weight. Then when you put in your unknown molecular weight protein, you can create a histogram of these binned particle impacts. And if you fit a Gaussian distribution to it, you can then get an average molecular weight for the protein that you're looking at. So as you can imagine, this would be very useful if you're trying to determine if something is going between a monomer and a dimer. So I was very excited to apply this to LPL. And my first theory for what was causing the dimerization in our CryoEM data was the presence of deoxycholate. And this was the bile salt that we use to break up the LPL elements and create a homogeneous sample for CryoEM. So I took LPL at a relatively low concentration, 16 nanomolar, and I put it on the mass photometer. And what you can see is that we get a single peak of around 54 kilodaltons when LPL is just diluted into PBS. And this matches up really well with the molecular weight of a single LPL monomer of 58.5 kilodaltons. So my next step was to mix my protein, LPL, with deoxycholate. And my theory was that what I would see would be a dimer of LPL appearing. However, that is not what I ended up finding. I found that the LPL, when I mixed it with deoxycholate, was still creating a single molecular weight peak, but that it was closer in molecular weight to a monomer than it was to a dimer. There is obviously a slight change here from 54 to 63 kilodaltons, which we believe might be from the association with the deoxycholate itself, changing the molecular weight. So this was a real surprise to me, and so I decided to test one more additive, which is known to interact with LPL, and that is heparin. LPL has 5 separate heparin binding sites on it. And when I mixed heparin with LPL at this concentration, I saw the same result, which is a single peak that most corresponds to a monomer of LPL. So at that point, I obviously needed to go back to the drawing board about what causes the dimerization. It wasn't actually the deoxycholate like I had thought it could be. So one of the things we knew from my work with the LPL filament is that concentration of LPL is important for filament creation. So essentially, this filament is concentration dependent. As you dilute LPL, you lose the filamentous form, and it predominates as these smaller LPL species. So we decided to see if increasing the LPL concentration could change what we were looking at with the oligomeric state. And so I used mass photometry again, increasing the concentration of LPL. And so this is LPL by itself diluted into PBS. And you can see that as I increase the concentration, we're seeing the appearance of a shoulder onto our monomer peak. This shoulder increases in size, and at our 100 nanomolar section, we see that we've shifted entirely to a molecular weight that corresponds most closely to a dimer of LPL at 99 kilodaltons. And so at least when I was looking at this, what I was thinking is that LPL is transitioning away from being only in the monomer state as I increase its concentration until a point where the dimer state predominates. So given that we froze our cryogrids at very high concentrations, this could potentially explain what we were seeing. One of the things that concerned me, though, was the appearance of yet another side peak at this 100 nanomolar LPL concentration, suggesting that potentially LPL would continue to adopt higher order oligomers as I increase concentration. Unfortunately, though, we weren't able to use mass photometry as we had done for these at higher concentrations, because as you can see, when you compare this 50 nanomolar LPL impacts to these 100 nanomolar LPL impacts, we were sort of maxing out our ability to determine individual particle impacts at 100 nanomolar. So we realized we needed to do something slightly different in order to characterize the oligomeric state of these higher concentration LPLs samples. And so, oh, sorry. I realized I forgot to show you guys the results with deoxycholate and heparin, and I hope you can see that they match what we saw with LPL by itself. So essentially, the deoxycholate and the heparin, neither of them were directly changing the oligomerization, as in both cases, you were going up to a higher concentration and you were seeing that it was trending towards the formation of a dimer. If anything, the deoxycholate sample was actually a little less likely to form a dimer at this concentration. You can see that this peak is still in equilibrium with the lower molecular weight peak. And that could be because of the existence of the deoxycholate in the sample, as detergent can affect how the particles impact the glass. But it certainly lets you know that the deoxycholate and heparin weren't the deciding factors as I had originally thought they could be. Right, so now back to my idea to use a higher concentration sample. And so the idea that I came up with was to use graphics. So this is a method where you have a gradient with increasing glycerol and cross-linker concentrations going through it. You layer your sample on top, and you can use a very high concentration sample. Then you spin it through an ultracentrifuge for 18 hours. And then when you take out your sample, you can fractionate it, and the sample has been cross-linked and separated by molecular weight. So this was a way to essentially assess the oligomeric state within a higher concentration sample, but now they've been separated out into these fractions where they're at lower concentration that I can then use mass photometry with. So just to give you an example of what these gradients looked like, if we took the fractions and we ran them on a Western blot, and here you can see we're blotting for LPL, we see the appearance of multiple oligomeric states, which I've indicated here with arrows. So we are certainly capturing potentially up to 4 or 5 oligomeric states. And interestingly, this red line here indicates where LPL would run if it were not cross-linked. And you can see that our lowest molecular weight sample of the cross-linked fraction is running below it. And that's because cross-linking actually changes the electrophoretic mobility of the sample. And so that's another reason mass photometry is going to be really useful for analyzing these, because it will be able to confirm the molecular weight of this sample that we're seeing here and confirm that it is a monomer of LPL as we suspect. So when we do the same thing but we use deoxycholate this time, we get the same distribution of different oligomers within our Western blot. So we were able to take the individual fractions from these graphics gradients and use mass photometry to analyze the oligomeric contents of them. And so for an example, I'm going to show you fraction number 9. So here's fraction number 9 from our Western blot. And here's what it looks like by mass photometry. So you can see that within this sample, we have monomers of LPL, dimers, trimers, and tetramers. So that was a real surprise to us that you were getting all of these oligomeric species, but it does kind of match with what we were seeing with the concentration dependence, as in when we increased the concentration of LPL, we were seeing the formation of dimers and the suggestion that you would even get further oligomeric structures, in this case, trimers and tetramers. And when we look at the deoxycholate sample, we see the exact same thing. So this obviously really drove home to me that the deoxycholate was not what was playing the crucial role in determining the oligomeric state. And indeed, you can see here the appearance of a small shoulder, suggesting that potentially we're also moving on to pentamers and even higher order oligomers in the presence of deoxycholate and just this, this increased concentration that we were able to use for the graphics gradients. So, at this point, I realized I needed to go back to the drawing board and really think critically about why LPL oligomerization differs between our CryoEM and solution studies, and try and determine a new hypothesis for what was causing this difference. And so I went all the way back to our original problem with the CryoEM, which was the preferred orientation. As I had mentioned when I was discussing it, when a protein adopts a preferred orientation in a cryogrid, it's because it's associating with the air-water interface. And so this sort of set off some light bulbs in my mind, because I realized that the air-water interface in some ways is very similar to the hydrophilic hydrophobic interface that LPL would interact with when it finds a triglyceride-rich lipoprotein. So in its association with the air-water interface, the hydrophobic residues of LPL were associating with this transfer from hydrophilicity to hydrophobicity. And indeed, that could explain why we saw the lid in an open position having undergone interfacial activation if LPL was interacting with this air-water interface as though it were a substrate mimic. So in solution, we see that LPL samples many different oligomeric states, monomers, dimers, trimers, tetramers, higher order oligomers. But then in the cryogrid, we see that LPL is favoring the dimer. And so my new theory is that the interaction with a substrate mimic, so a large-scale substrate mimic, the one that mimics the interface that LPL interacts with of a triglyceride-rich lipoprotein, is what triggered the formation of this dimer. And that when we're looking at LPL in solution, when, even in the presence of detergent like deoxycholate, we're not seeing it alter the oligomeric states because there may be something fundamentally different about a small-scale substrate mimic, so sort of a soluble substrate mimic, compared to this large-scale, interface substrate mimic. And, this opens the questions of, can these small substrate mimics, these soluble substrate mimics, cause interfacial activation? Can they cause different forms of dimers to populate? That is a question that we're still going to have to keep investigating to answer. But I do think that this theory explains potentially why we see the pore opening in our structure. So in association with the air-water interface, LPL has now exposed the active site to a very hydrophobic air environment. And that could be what triggered the opening of this pore. Whereas if you had LPL in a soluble environment where you have hydrophilic interaction with the LPL active site, you're not going to favor the opening of a hydrophobic pore. In fact, it would be extremely disfavored because it'd be better for that hydrophobic pore to stay closed, as that would be a lot more energetically favorable. However, if a triglyceride-rich substrate then can act, the air-water interface then can act as a mimic potentially for this triglyceride-rich substrate in terms of opening of the pore. And so to kind of finish off for the day, I kind of want to come back to what we're thinking in terms of the LPL dimer. And so what our current theory is that association with this triglyceride-rich substrate interface is what's triggering LPL to form the active dimer. This active dimer associates its hydrophobic residues with the hydrophobic triglyceride-rich lipoprotein, and this interface is what triggers the opening of the lid peptide, so that's interfacial activation. And it's also going to trigger the opening of the pore because it's exposing the pore residues to a hydrophobic environment, which would allow them to open. This then would allow LPL substrate, the triglyceride, to bind. And we think that there are more substrate discrimination sites here than we had previously thought. It had been previously thought that this small pocket up at the top was how LPL discriminated between different substrates. But it seems perhaps that there's also a hydrophobic pore and then a secondary hydrophobic pocket for the SN2 chain that can also help discriminate to make sure that LPL is binding to the right substrate, specifically the triglycerides. After it binds to the acyl chain, this then allows water to come in and hydrolyze that bond that links the acyl chain to the glycerol backbone. And this then liberates a free fatty acid. And our theory is that this free fatty acid could then travel unidirectionally out of the pore and into the capillary, where it would then interact with albumin for transport across the capillary endothelium. And this is a very, I guess, appealing idea because it would be very processive, because it would mean that LPL wouldn't have to dissociate from the triglyceride-rich lipoprotein in order to release the free fatty acid, which would mean it wouldn't have to re-undergo interfacial activation every time. Another thing that I'm currently interested in looking at is whether or not this secondary pocket for the SN2 chain could allow processive hydrolysis of the SN1 and SN3 chains of a single triglyceride. So essentially going from a triglyceride to two free fatty acids and a monoglyceride, rather than going to a diglyceride with one free fatty acid. So those are areas of future investigation in the near lab. So our model right now for the LPL homodimer in the body is that it exists when LPL is in the capillary and in association with triglyceride-rich lipoproteins. So as I mentioned earlier, we know that GPI-HBP1 is what brings LPL into the capillary, and it anchors LPL to the capillary wall. There's also heparin sulfate proteoglycan that also line this the capillary wall as well, that could also anchor LPL. When LPL interacts with a chylomicron, you begin to get the production of free fatty acids. And it has been shown that when you have LPL, GPI-HBP1 complexes, introduction of substrate, such as chylomicrons, or product, such as free fatty acids, actually triggers the release of LPL from these GPI-HBP1. And so our theory is that after that release is when you would then form the LPL dimer. This would allow LPL to continue to be active on this protein by orienting its active sites and its hydrophobic interaction sites with the actual chylomicron in a way that is mutually exclusive to the GPI-HBP1. And as I mentioned briefly earlier, lots of previous research has shown that the final role of the LPL homodimer will be in traveling with the lipoprotein remnant to the liver. Once the remnant has decreased in size, that means you've hydrolyzed most of the triglycerides and actually changed the size of the remnant, it can then pass into the space of DISI and interact with LRP, which this homodimer facilitates, and that allows the remnant uptake into the liver, where it can then be repackaged into other triglyceride or into other lipoproteins. So to come back to the summary slide I started with, as you can see, LPL has a variety of different structural transformations throughout its life, and it's highly regulated at a variety of steps. And what I've talked to you about today is mostly this LPL homodimer step. But what I hope you can appreciate is that all the evidence that we have now from our solution data with LPL suggests that there's further oligomeric states that we have not structurally characterized yet. And where those states might exist, for example, in the interstitial space, is something that really is very interesting and remains to be explored. So still lots to figure out here. And with that, I would like to thank my mentor, Saskia Neer, everyone in the Neer lab, Brian Coleman, who helped me with the Rosetta modeling, my sources of funding, and of course the UNC core facilities, particularly the CryoEM facilities, where Josh Strauss and Jared Peck helped with the collection of data for solving the CryoEM structure, and the macromolecular crystallography core and its director, Nate Nicely, where the mass photometer is housed at UNC. And I also wanted to let you guys know that I'm actually going to be leaving UNC in January, and I will be opening my own lab at Stony Brook University. And we are actually going to be continuing studying how quaternary structure and oligomeric state of lipases can influence their activity. And with that, I would love to take some questions.